Hi everyone, welcome to my channel Dr. Venu Madhav's Learning Anatomy and Physiology. Today we are going to discuss about the basic design of the animal cells and a few functions of the important organelles of the cell. First of all, what is cell? The cell is the basic unit of life and all living organisms are composed of many units called cells. Each cell has all the characters of the life. A cell can be defined as the basic living structural and functional unit of the body. That is the definition given to the cell. A cell can be defined as basic living structural and functional unit of the body, and the study of the cells is known as cytology. If we see the cell surroundings, the extracellular space is the space outside the cells and is of two types: the interstitial space between the cells of a tissue and the vascular space outside the cells in blood vessels. The extracellular fluid includes all the fluids present outside the cells like interstitial fluid, plasma, transcellular fluid, etc. The extracellular matrix, it is the complex meshwork of components secreted by the cells into the interstitial spaces. These include the glycoproteins, proteoglycans and fibers like collagen, elastin, etc. If we see the general characteristics of a cell, each cell needs nutrition and oxygen. It produces its energy required for growth, repair and other activities on its own. Excretion of metabolic waste and carbon dioxide has to be done, and cell has to maintain its internal environment. The cell shows response to the invasion of bacteria and toxic substances, and it reproduces by division, thus maintaining its offsprings. And after division, it may die. That is, sometimes it is programmed or sometimes due to the other factors. The programmed cell death is apoptosis. That means the cell has to undergo all the stages in the life of an organism. So, it is called a basic structural and functional unit of life. The cells usually exists as groups or sheets in human body. The side of the cell that faces either lumen or lumen of the organ or the external surface is called its apex, and the side which is present opposite to the apex is called base of the cell. Other two sides are called lateral surfaces. The shape of the cell varies depending on the function of the cell. The apex, base, etc. are confined to the fixed cells, and the shape of this cell also confined to the fixed type of cells. For example, in protective layers, the cells may be stratified squamous. The lowest layer is on the basement membrane, it is fixed there, and where diffusion is needed, it is simple squamous epithelium. The cell may be cuboid in shape in lining the ducts, and columnar in cells where secretion is the main function. Secretion or absorption, it should be the columnar. If we see the structure of an animal cell, in general shows many features and all can be grouped as following parts for our understanding. The plasma membrane, it is the outer covering of the cell which separates the external environment from the interior of the cell. The cytosol is the thick semi-fluid which is present within the cell. It contains the dissolved proteins, nutrients, enzymes, ions, etc. And all organelles and the organelles of the cell and inclusions are present in this cytosol. And the cell organelles are highly organized structures with characteristic shapes that are specialized for specific cellular function. Largest of organelles is the nucleus. There are inclusions in the cytosol. Those are the temporary structures that contain secretions and storage products of the cell. The cytoplasm includes the cytosol, all the organelles except the nucleus and the inclusions. This is the animal cell what we are seeing here. You can see number of organelles like the nucleus, peroxisome, centrosome, centriole, lysosome, ribosomes, cilium, etc. So, we will see one by one in detail. We will start with the plasma membrane or the cell membrane. The plasma membrane is the thin layer that separates the internal components of a cell from the extracellular materials which are present around the cell. So, this is also called as plasma lemma, and this layer helps in protection of the cell and gives specific shape of the cell. This layer regulates the passage of the materials into and out of the cell, which is called permeability, which is selective for specific materials. If we if we see the structure of the cell membrane, on the basis of the structure, the cell membrane is called unit membrane, and various scientists described various structural models of cell membrane, out of which the Danieli-Davison model is the first proposed model, and it described cell membrane as a sandwich of lipids covered by proteins on both sides. And unit membrane model, that was explained by the Robertson on the basis of electron microscopic studies, and another one is the fluid mosaic model, that is proposed by the Singer and Nicholson. This model is more acceptable by many. You can see the fluid mosaic model. According to this, the cell membrane is a fluid which have the mosaic of proteins. In this model, the proteins are found floating in a fluid of lipids. The plasma membrane consists of proteins and lipids in a 50-50 ratio by weight. Proteins are larger in size and heavier than the lipids, so their number is less, and all these are held together by non-covalent bonds. If we see the structure of the lipid layer, the plasma membrane consists mainly of phospholipids which are in fluid form. This moves from one place to another along the surface of the cell. The lipids present in the membrane are mainly phospholipids about 75% and glycolipids about 5%, and the cholesterol, that is forming about 20% of the lipids that are present in the plasma membrane. The bilayer forms the basic framework of the plasma membrane. The glycoproteins and the glycolipids form the glycocalyx over the cell surface. The phospholipids, if you see, the phospholipids are arranged in two parallel layers which forms a phospholipid bilayer. The phospholipids are amphipathic in nature, that means they have both polar and non-polar regions in the same molecule. The polar part contains phosphate and is the head of that molecule and is hydrophilic. Hydro means water and philia means loving, so they attract water. And the non-polar part has two fatty acid tails which are hydrophobic, they hate water. If you see the arrangement of the phospholipids in the bilayer are arranged in a specific manner with the hydrophilic heads towards the outer and inner surfaces of the cell membrane. That means the outer phospholipids face the external environment and the internal phospholipids face the internal environment, that is the cytosol. The hydrophobic tails of these lipids are present within the inner aspect of the membrane facing each other. This is how they are arranged. You can see the hydrophilic end, that is the hydrophilic head towards the extracellular fluid, and the hydrophobic tails of the lipid bilayer are facing each other. Now, the other lipids of the cell membrane, if we see the glycolipids, these account for the 5% of the membrane lipids and usually appear in the layer that is towards the extracellular fluid. Useful for adhesion among the cells, may mediate cell to cell communication and may contribute to the cell growth and development. Another type of lipids is the cholesterol, contributes to the rest of the lipids of the cell membrane located among the phospholipids, useful in strengthening of the cell membrane. If we see the functions of a lipid layer, the lipid layer is semi-permeable membrane, allows fat soluble substances only to pass through. Fat soluble substances like oxygen, carbon dioxide and alcohol can pass through the lipid layer easily, but the fat insoluble substances like glucose and electrolytes cannot pass through this layer. If we see them if we see the membrane proteins, there are two types of proteins in the cell membrane: integral proteins and peripheral proteins. The integral proteins, these extend across entire thickness of the phospholipid bilayer, and they are also named as transmembrane proteins. So, these are tightly bound with the cell membranes. Most of these proteins are glycoproteins attached with the sugar groups. If we see the integral or intrinsic transmembrane proteins, they cell adhesion proteins these are, and some of these proteins have a pore through them which act as a channel through which certain substances can flow into and out of the cell. Few cells act as transporters to move a substance from one side to the membrane to the other side of the membrane. Few act as receptors to the hormones, neurotransmitters, and are specific to specific substances, and these are named as ligands. If we see the peripheral proteins or extrinsic proteins, these do not extend across the phospholipid bilayer. They are loosely attached to the inner and the outer surfaces of the bilayer and can be easily detached from the membrane. These proteins along with integral proteins serve as cytoskeleton anchors. They form attachment between plasma membrane and the cytoskeleton of the cell. If we see the types of the peripheral proteins, there are two types: intracellular peripheral proteins, that means they are present on the inner aspect of the cell membrane, and these include receptors, transport proteins, and some enzymes. The extracellular peripheral proteins, they are situated on the outer surface of the cell membrane. They include antigens, adhesion proteins, and some of the enzymes. You can see the function of proteins in cell membranes. The integral proteins provide structural integrity of the cell membrane. Channel proteins help in diffusion of water soluble substances like glucose and electrolytes. Carrier proteins transport certain substances across the cell membrane by means of transport mechanisms, and receptor proteins act as receptors for hormones and neurotransmitters, some of the enzymes. Some of the proteins form enzymes also. Some act as antigens and induce antibody formation, and some act as cell adhesion molecules forming intracellular junctions. The membrane glycoproteins and glycolipids often are called the cell identity markers. They may enable a cell to recognize other cells of its own kind during tissue formation. They also recognize and respond to the potentially dangerous foreign cells. Examples are the ABO blood type markers. They identify the opposite blood group which is harmful for our blood group. This is the cell membrane, how it appears. You can see the lipid bilayer. Heads are towards the outer side and tails are in the center, and you can see a channel protein, peripheral protein, integral protein, and cholesterol, glycolipids, everything are there in this image. You can see if you see the cytosol is the intracellular fluid in which organelles and inclusions are suspended and the solutes are dissolved. Cytosol is a viscous transparent gel like fluid. The cytosol consists of microfilaments and microtubules which form the cytoskeleton of the cell. Cytosol is made up of 70 to 90% of water, in which solid like proteins, carbohydrates, lipids, few amino acids, and inorganic salts are present. The cytosol is the medium in which many metabolic reactions occur. If we see the organelles, organelles means the little organs. They are the specialized structures that have characteristic appearances and specific role in growth, maintenance, repair, and control. Their number vary from cell to cell. Some important organelles we all know: the nucleus, ribosomes, endoplasmic reticulum, Golgi complex, lysosomes, peroxisomes, mitochondria, centrosomes, and centrioles. If we see, we start with the nucleus, that is called kernel. Nucleus is the most important structure in a cell. Just like the kernel in a seed, the nucleus is the most important structure in a cell. So, nucleus is seen in all cells except RBC. It is a round or oval structure usually present in the center of the cell. Nucleus consists of a viscous fluid called nucleoplasm. It is covered by a nuclear envelope which has small pores in it, consists of genetic material in the form of chromatin, that is the DNA, and the chromatin fibers will condense to form chromosomes during cell division. The uncoiled chromatin in normal sections is called euchromatin, and coiled part of the chromatin is called heterochromatin. You can see this is the nucleus, and you can see the envelope having many pores, the chromatin is there, the chromosomes are there, and another structure, the small rounded nucleolus, is also seen within the nuclear envelope. A plano-convex heterochromatic body is seen under the nuclear envelope in normal females. This is the inactive X chromosome and is called bar body or the sex chromatin. If we see the functions of the nucleus, it is the most important organelle of a cell, as it controls all the cell activities including metabolism, protein synthesis, growth and reproduction. So the nucleus synthesizes the ribonucleic acid, and it helps in the its function is the formation of subunits of ribosomes and sends information to the cytoplasm for protein synthesis through the messenger RNA. It controls cell division with the help of genes, stores genetic information in genes and transfers the information to the next generation of cells and organism. So these are the main functions. If we see the ribosome, the name ribosomes is due to the presence of the ribonucleic acid, that is the RNA. The ribosomes are tiny granules that contains ribosomal RNA and ribosomal proteins. The rRNA is synthesized by the DNA in the nucleolus, and ribosomes consist of two subunits, one about the half of the other unit, and ribosomes are the site of the protein synthesis, and they usually found attached with the rough endoplasmic reticulum. You can see there are two subunits, and they sometimes the ribosomes may float. They are called free ribosomes. Now the endoplasmic reticulum. The endoplasmic reticulum is a network of membrane enclosed channels of varying shapes. Those are called cisterns. These cisterns are interconnected with each other, and the outer surface is formed by proteins and lipid bilayer. Lumen is filled with the endoplasmic matrix, and this endoplasmic reticulum is continuous with the nuclear envelope. It is present very close to the nucleus, and you can see the types of endoplasmic reticulum. There are two types. There is a rough endoplasmic reticulum and smooth endoplasmic reticulum. The rough endoplasmic reticulum is studded with the granular ribosomes and its functions is the synthesis of proteins. With the help of the ribosome, they produce glycoproteins. Degradation of worn out organelles, this is one of the important functions. The rough endoplasmic reticulum wraps around the degraded or the worn out organelles and forms phagocytic vesicles. Later they are phagocytized by the lysosomes. And now you can see the another type is the smooth endoplasmic reticulum. The smooth endoplasmic reticulum is devoid of ribosomes and appears smooth. Contains interconnected smooth tubules, and its functions is synthesis of fatty acids, phospholipids and steroids. It plays an important role in metabolism with the enzymes present in the reticulum. Major site of storage and metabolism of calcium. The smooth endoplasmic reticulum involves in catabolism and detoxification of toxic substances like certain drugs and steroids. You can see the Golgi apparatus. These are located near nucleus, more extensive in the cell with high secretory activity. Consists of flattened sacs called cisterns. The cisternae are stacked upon each other like pile of flat plates. Those which bulge at their edges, and small vesicles called Golgi vesicles are present around these expanded edges of the cisterns. The Golgi apparatus has two faces. The cis face is positioned near rough endoplasmic reticulum because the reticular vesicles enter Golgi apparatus through this face. The vesicles which are produced by the endoplasmic reticulum, they enter Golgi apparatus. Then transpace, that is positioned cell near the cell membrane, that is the plasmalemma, and the processed substances exit through this surface. The Golgi complex processes, sorts and packages and also transfers the proteins and lipids to the cell membrane. Also forms lysosomes and secretory vesicles. This is the Golgi apparatus. You can see the cis face on one side and the transpace on the other side. Lysosomes are the membrane-bound vesicles formed in the Golgi complex. They enclose variety of hydrolytic enzymes which are active in acidic pH. The membranes of lysosomes has proton pump which drives the hydrogen ions into the vesicle and to maintain the acidic pH of about 6.5. The lysosomes are the vacuoles filled by the hydrolytic enzymes. There are primary lysosomes, they are freshly formed the Golgi apparatus and are inactive. The secondary lysosomes are the active form of the lysosomes, and we can see a few enzymes in some lysosomes. Those are the proteases which are hydrolyzes the proteins into amino acids, lipases which hydrolyzes lipids into fatty acids, amylases hydrolyzes polysaccharides into glucose, and nucleases hydrolyzes the nucleic acids into mononucleases. If you see the functions of the lysosomes, degradation of macromolecules is done by the lysosomes because they are useful for phagocytosis by engulfing and digesting the larger molecules like bacteria and other substances that enter the cell, and the degradation of the worn out of the organelles. Lysosomes also help in recycling the cell structures. They can engulf other organelles, digest and release the digested materials into cytosols for reuse. This is called autophagy. Removal of excessive secretions. Liquids are engulfed by lysosomes by the pinocytosis and engulf endosomes in endocytosis. Autophagocytosis. The lysosomes may destroy their host cell. Process is called autolysis. Usually occur after death of that cell. Secretory lysosomes secrete certain substances like perforins and granzymes which act as killers of viruses. Melanin and serotonin also secreted by few lysosomes. If you see the peroxisomes, these are similar to lysosomes but a small structure. Pinched off from the endoplasmic reticulum, they contain oxidizing enzymes like catalase and oxidases. They break down fatty acids by means of beta oxidation. They degrade toxic substances like hydrogen peroxide by means of detoxification and forms the major site of oxygen utilization of a cell. They accelerate gluconeogenesis from the fats, degrade purine into uric acid. They participate in formation of myelin and bile acids. So, peroxisomes are also very important. The most important is the mitochondria. Single is mitochondrion is known as the powerhouse of the cell, as these are the main sources for the generation of energy. These are oval or rod shaped, consists of two membranes similar to the cell membrane. The outer layer is very smooth and it covers the contents of the mitochondria, but the inner layer forms shelf-like inward folds called cristae. The central cavity of the mitochondrion that is enclosed by the inner membrane and the cristae is called the matrix. Surface on the cristae is the site for cellular respiration where the ATP is generated. The ATP is the adenosine triphosphate which is the power of the cell is generated. If you see the functions of mitochondrion that produces energy, the energy is produced during oxidation of proteins, carbohydrates, etc. by the oxidative enzymes on the cristae. During this process, the water and carbon dioxide are released along with energy. This energy is stored in mitochondrion and later used for the synthesis of ATP. Synthesis of adenosine triphosphate. The components of respiratory chain in the mitochondria are responsible for the synthesis of ATP by utilizing the energy released during oxidative phosphorylation. The apoptosis, the cytochrome C of the mitochondrion involved in apoptosis, that is the programmed cell death. Storage of calcium in matrix is there and detoxification of ammonia in the mitochondria of the liver cells. So, these are the functions of the mitochondrion. If this is the mitochondrion, you can see the inner membrane, the outer membrane, inner membrane forming the cristae and the matrix is there. So, now you coming to the cytoskeleton part, the shape of the cell and its movements and also the movements of the internal organelles are due to the presence of fine filaments of protein within the cytosol, which forms the cytoskeleton of the cell. Cytoskeleton is useful in phagocytosis, movement of organelles during cell division, etc. three main type of protein filaments are seen in cytoskeleton: the microfilaments, microtubules, and intermediate filaments. You can see the cytoskeleton components. The microfilaments, these are the rod-like structures formed from the protein actin. In a muscle cell, these filaments produce muscle contraction. In a non-muscle cell, these provide support and assist in cell movements, secretion, and phagocytosis. The microtubules, these are larger than microfilaments, straighter and slender filaments. These contain a protein called tubulin. These assist to provide shape to the cell and also to move organelles within the cell. These microtubules are more around the centrosomes. The intermediate filaments, size is the intermediate to the above the two filaments, but stronger than the above. These filaments reinforce the shape and structure of the cell. These are examples are keratins in epithelia, glial filaments in astrocytes, neurofilaments in nervous tissue, and desmin in muscles. The centrosomes and centrioles. The centrosome is a dense area of cytoplasmic material with radiating microtubules present near nucleus. The centrosomes are the centers for the organizing of microtubules in non-dividing cells and form mitotic spindles during cell division. Within the centriole are a pair of centrioles which are arranged at right angles to each other. Each centriole is composed of 9 clusters of 3 microfilaments arranged circularly. The centrioles play a role in the formation of flagella and cilia. You can see the centrosomes are there. You can see the two centrioles are arranged at right angles to each other. The flagella and cilia, some cells have projections which help in movement of the cell or the substances along the surface of the cells. If the projection is long and useful for the movement of the cell, these are called flagella. Example, flagellum in humans is the tail of the sperm cell. If the projections are numerous and short, projecting from the surface of the cell as hair, they are called cilia. They help in moving the substances along the surface of the cell. Example, the cilia in the respiratory tract, which drives the dust particles etc. outwards. The flagella and cilia consist 9 pairs of microtubules that form a ring around a central pair of microtubules in the center. Both also contain cytoplasm and covered by the plasma membrane. If we see the cell inclusions, the inclusions are large group of chemical molecules produced by the cells. They may appear or disappear at various stages. The inclusions include melanin pigment in the skin, glycogen in the liver, triglycerides etc. And also few steroids, enzymes. These are all inclusions. With this, we conclude here at the cell. The cell is very important for the life, and it is the basic structural and functional unit of our life. So, please subscribe to my channel and watch the video completely, and please click the like button so that it helps in promoting my videos. And also please subscribe and click the bell icon to get the latest update of the videos. Thank you very much.